Today we will discuss about chapter number 2, 11th class, biological classification. Introduction. Aristotle was earlier to attempt a more scientific basis of classification. He used morphological characters to classify the plants into tree, shrub and herb. So it is very important. Sometimes they'll ask the questions. Aristotle first he took the morphological character or sexual characters or other characters. The answer will be come to point, that is morphological characters. He also divided the animals into two groups. Who? Aristotle. Red blood group of animals, he given the term anemia, and without red blood groups they gave the name is called anaima. So the two kingdom system of classification was done by another scientist, the Carlos Linnaeus. He divided the total kingdom into plants and animals, two groups only. Next, a three kingdom system of classification was done by Ernest Haeckel. He was a German zoologist in 1866. Next, four kingdom classifications done by Copeland in 1956. He just divided the Mycota, Protista, Plantae, Animalia. So here remember that Mycota is nothing but Monera. Next, five kingdom classification was done by R.H. Whitaker in 1969, and he just divides the total living things into Monera, Protista, Fungi, Plantae and Animalia. And last, the sixth kingdom classification done by Carl Oz, Candler and Willis. So they divide the Archaeobacteria, Eubacteria, Protista, Fungi, Plantae and Animalia. That means very simple. So in case of the first, they divide the Monera into just two groups, especially Archaeobacteria and Eubacteria. Next, the objection came to the Linnaeus. Sometimes they will ask the questions, what are the objection or demerits of Linnaeus? Means the first point, he was not distinguished between eukaryotic and prokaryotic cells. Second, he was not distinguished between unicellular and multicellular, and he was not distinguished between the photosynthetic and non-photosynthetic characters. So last option they given all the above. That means you can find out that answer is all the above. Next, main criteria for classification used by Whitaker. So nowadays we are considering the five kingdom classification done by Whitaker. So he include who? Whitaker include the cell structure, thallus organization, mode of nutrition, reproduction, phylogenetic relationship. So what the meaning of phylogenetic relationship means? The evolutionary trait. So these are the characters include who? Whitaker. And sometimes they will ask the question for this. So main criteria of classification used by Whitaker means you just find out that the correct answer. Correct answer means either cell structure or mode of nutrition, reproduction, thallus organization or phylogenetic relationship. Any one you can select if it is find out the correct answer means. Next, you just find the Monera include all unicellular prokaryotic cells. All of you know that about the prokaryotic cells and eukaryotic cells. So especially we are just prokaryotic cells means we will give the example is bacteria. Next, Protista. Protista include all unicellular eukaryotic cells. So eukaryotic cells means that is you can take the plants, higher plants, animals, all are undergo the eukaryotic cells. Now previously what happens, the Chlamydomonas and Chlorella earlierly placed in the algae, but these two group undergo the unicellular, so they consider as the unicellular eukaryotic cells undergo the Protista. Like that animals are there, Paramecium and amoeba, Earlier they kept in the animals, but these two are unicellular eukaryotic cells, so they include nowadays undergo that kingdom Protista. So let it be think the first kingdom is Monera. So kingdom Monera. In this kingdom, they just include few groups like Archaeobacteria, Eubacteria, Mycoplasma, and Actinomycetes. First of all, we will discuss about that Archaeobacteria. So Archaeobacteria nothing but it a group of ancient bacteria. Some points you can note down. These points are not available in the NCERT books. Let's come to the points. Habit. What is the habit of Archaeobacteria? That means Archaeobacteria are live in the most harsh habit, especially that is salty area we call as holophilis, or hot spring that is called thermoacidophilis. That is means they can survive in up to 80 degrees Celsius and pH 2. So now here we discuss that all of you know that enzymes can be denatured if the temperature is increased more than 45 degrees Celsius. But in the Archaeobacteria, the enzymes, those enzymes are present in Archaeobacteria. These enzymes can survive more than 45 degrees Celsius. They can't destroy or denature up to 80 degree or more than 80 degrees Celsius. So now these Archaeobacteria are oldest living fossils. It is very important points. You just note down and remember this. Archaeobacteria are oldest living fossils, and next they are mostly obligate anaerobic bacteria, or you can say obligate anaerobes. Sometimes they will ask the questions. So find out the example of obligate anaerobes. So obligate anaerobes means you can take the Archaeobacteria. Here the few characters are there. These characters are not found in the general bacteria or Eubacteria. So here cell wall of Archaeobacteria is differ from other bacteria in having pseudomurine instead of peptidoglycan. So peptidoglycans are found in the cell wall of general bacteria, but in Archaeobacteria it is instead of peptidoglycan, the another chemicals is that is called pseudomurines. And these bacteria also they can live in marshy area, especially called methanogen bacteria, we can say that. And you come to the cell membrane. Cell membrane contain branches of a chain of lipids, especially you know the plasma membrane. It is mainly constitute of or consist of lipids and protein, so that's why it is called a unit membrane. But here what happens in Archaeobacteria, the cell membrane contains branch of chain lipids only, proteins are absent. And next, the methanogens are also present in the gut of several ruminant animals like cow and buffaloes, and they produce a gas that is called methane gas. Next, some structures are there here. The structure of bacteria according to that the shape, and the extra points you note down that here very important. The Barophilic Prokaryotics, remember this point. Barophilic Prokaryotics are prokaryotics which grow and multiply in very deep marine sediments. So these bacteria, Archaeobacteria, are found in the deep marine sediments, we called as Barophilic Prokaryotics. Next, the Eubacteria.